So thank you for that introduction. As Manis mentioned, my name is Manisha, and I am a postdoc at the University of Minnesota, where I've worked with Drs. Nathan Springer, Yaniv Bronvain, and Dr. Candy Hirsch. And today I'm going to be talking about a project I've been working on that's centered on exploring how promoter structural variation influences genome-wide expression patterns in domestic maize, and how this relates to transposable element content. And with that, I'm just going to go ahead and get started. So a basic question in genetics is understanding and exploring how present or observed genetic variation impacts organismal phenotypes. And to that end, we know that genetic variation is in itself variable. It can be as simple as a single nucleotide change at a specific base pair, or a small insertion or deletion event that slightly shifts the genetic sequence. We also know that there can be much larger structural variants that introduce sizable structural variation into the genome. The range of sizes for structural variants, often referred to as SVs, is broad, with some just greater than 50 base pairs, while others contain several megabases of sequence. Attempts to characterize the overall amount of variation within a genome attributable to any of these different types estimate that SVs are introducing as much, if not substantially more, base pair variation relative to SNPs. But they've been comparatively understudied, and this is due in part to technical limitations. Short-read sequencing approaches have really struggled to accurately identify SVs, due in part to their size and the strong association between SVs and repetitive regions. One such association is due to the presence of transposable elements. Transposable elements, or TEs, are selfish genetic elements that are capable of copying themselves and inserting into new positions within the genome. And this consequently generates structural variation at this new insertion site. TE-mediated structural variation is unique for two different reasons. First, TEs contain enhancers, promoters, and other regulatory features in order to recruit the cellular machinery required for their transposition. So TE-mediated SVs specifically introduce novel regulatory features at their insertion site. Additionally, TEs are often silenced via epigenetic modifications, such as DNA methylation, histone modifications, or small non-coding RNAs. And these epigenetic modifications are often imprecise in that they can spread from their initial deposition site, and dynamic in that they shift in cross-cellular contexts or in response to environmental stimuli. So in addition to introducing regulatory sequences, TE-mediated SVs can also change the epigenetic landscape near an insertion site. And these two features of TE-mediated SVs present an interesting opportunity for shaping gene expression patterns. To break this down, I like to think of a single gene in a specific context that has some amount of expression. Agnostic to its sequence, a structural variant within the promoter region could change gene expression patterns simply by changing the distance between regulatory elements and the gene of interest. This can either increase or decrease expression depending on the nature of the regulatory element itself. Now, if this SV is TE-mediated, we could expect increased expression of the nearby gene if the internal regulatory features housed within the TE inadvertently influence expression of the downstream gene. On the other hand, if this TE is epigenetically silenced, which it usually is, we might actually expect decreased expression instead. So here, the epigenetic modifications used to silence the TE might spread into associated regulatory features or even the gene itself. And furthermore, because epigenetic modifications are dynamic, we might actually observe both of these states across different contexts, or potentially even transition between them in response to environmental stimuli. So there's a variety of ways with which an SV within within the promoter region of a gene could influence expression patterns. And in fact, an increasing number of studies are finding that SVs associated with phenotypic differences are often found in non-coding regulatory regions like the promoter. That being said, it's still unclear how often and in what ways SVs change gene expression patterns across the genome. So are promoter SVs associated with genome-wide changes in expression patterns, or are these really limited to a handful of specific examples within the genome? When we do observe changes in expression, are these differences occurring across all tissues, or are they very context-dependent? And for promoter SVs that are associated with differential expression, are these changes being caused by any similar feature of the SV, or do these represent more random stochastic changes in expression? And finally, do we actually observe any special role for TE-mediated SVs in changing gene expression patterns, or do they behave similarly to other promoter SVs? So to actually evaluate these questions, we decided to use domestic maize, as over 70% of its genome has been annotated as TE sequence, making it an ideal study system for this question. In particular, we decided to use a collection of 26 diverse inbred lines that represent the breadth of maize genetic diversity. These lines are a really powerful pangenomic resource with a whole collection of publicly available datasets, including genome assemblies, TE annotations, and extensive transcriptomic data, which is essentially everything we need to study this question. From these genome assemblies, we had already identified SVs across these panel of lines using pairwise whole genome alignments generated using anchor wave. And we had actually taken these SVs and already intersected them with TE annotations. So we not only know the location of all SVs across this panel of lines, but we also know whether and how they contain TE sequence. In order to test whether promoter structural variation is associated with differential expression, we actually had to identify genes that had structural variation within the promoter. And so here, the maize reference genome B73 happened to be one of our 26 inbred lines. And for each B73 gene, genotypes sharing the same SVs across the promoter and gene body were grouped into structural haplotypes. We then identified genes that had at least two structural haplotypes, one that must be B73-like. You can think of this as our no SV state, and at least one where the only structural variation is within the promoter, so no structural variation within the gene body. We also required that each of these structural haplotypes be represented by at least 3 inbred lines in order to give us the statistical power to test for differential expression. This left us with about 12,000 genes, which represented about 30% of the full gene set. For each inbred line, we have transcriptomic data for 10 different tissues taken at various developmental stages. And we could use these two data sets to test whether there is differential expression between structural haplotypes and in which tissues we observe differences. Now, if you've been listening really closely, you might have clocked that we define structural haplotypes exclusively using SV content. But it's certainly possible that there's another variant co-segregating with these structural haplotypes that's actually causal for the differential expression. So when we test for differential expression, we're going to find it, even though the SV isn't necessarily what's causing it. But for time, I didn't have time to go into this. We did look at some examples where we looked at promoter SNP content and looked to see how often it was segregating by our structural haplotypes, and we did not find this happening very frequently. And so we don't believe this is a pervasive issue across our candidate gene set, but therefore it's not really appropriate for us to look at any one specific gene and be like, this SV causes differential expression. But it is okay to look across all of our differentially expressed genes and their associated promoter SVs and see if any patterns emerge. So we're really looking at these differentially expressed genes in aggregate. Okay, so how often are promoter SVs associated with differential expression? So using our model, we find about 3,500 candidate genes that show differential expression between structural haplotypes. This is about 30% of our candidate gene set and about 8% of all B73 genes, which is pretty substantial. To evaluate whether this level of differential expression exceeds what we would expect by chance, we constructed a random mimic of our data set. Basically, for each candidate gene, we randomly selected a non-tested B73 gene and assigned it the same haplotype structure as one of our candidate genes, but retained its original expression values. And then we just used the same differential expression model to test for DE between these artificial structural haplotypes. And for this random mimic of our candidate gene set, we found only 639 differentially expressed genes, which is nearly a 5.5-fold reduction in the number of DE genes. And so this suggests to us at least that promoter SVs are associated with differential expression more frequently than expected by random chance. So we do believe that promoter SVs are associated with differential expression and that this is occurring broadly across the genome, as over 30% of our tested genes exhibited DE. We then wanted to explore whether this differential expression was consistent across our tested tissues or if it was specifically occurring within a subset of tissues. To do this, we took a tissue-by-tissue approach, where we basically just tested whether a candidate gene was differentially expressed between structural haplotypes within that tissue. And then we could then classify genes based on how many tissues we observed a differential expression within. 27% of our candidate genes were never differentially expressed in any tissue. And only 2% of our candidate genes exhibited differential expression across all 10 tissues. In fact, the vast majority of our candidate genes exhibited some amount of tissue-specific differential expression. We were curious whether these genes that were differentially expressed but not across all tissues were because they lost differential expression in one or two tissues, or if they were really being driven by a subset of small subset of tissues. And so for our tissue-specific analysis, we didn't test a gene if it wasn't expressed within that tissue, so we know that that's not causing this loss of differential expression. So what we did is we looked at our subset of candidate genes that were expressed across all 10 tissues and basically counted the number of tissues we observed differential expression in and plotted out this distribution. And what we found was that most genes were showing differential expression in anywhere from 1 to 5 tissues. So it really did appear to be tissue-specific versus broad expression changes that were maybe missing in 1 or 2 tissues. So yes, the vast majority of genes that are associated with differential expression between structural haplotypes exhibit these tissue-specific differences or these context-dependent differences. Okay, so we now wanted to explore whether there were any similar features of promoter SVs that were associated with differential expression or of the genes themselves. We looked at a lot of different things. I'm just going to give some of the more relevant ones. But basically, we started by looking at the size of the SV and how close the SV was to the transcription start site, as we hypothesized that larger SVs, or potentially those that were closer to the TSS, might be more likely to disrupt associations between regulatory features and the gene of interest. But we found no difference between promoter SVs that were associated with differential expression and those that were not associated with differential expression. We also looked at the frequency of the promoter SV within the population, as we hypothesized that perhaps rarer SVs would be more likely to impact expression. But once again, we found no difference between SVs that were associated with DE and those that were not. We then shifted to looking at the genes themselves. We hypothesized that perhaps genes with lower expression or those that were expressed in fewer tissues might be more likely to exhibit differential expression. Once again, no difference between SVs that are associated with genes that had DE and genes that did not have DE. We looked at the evolutionary conservation / age of the gene by looking at both the pangenome and phylostrata classification of differentially expressed versus non-differentially expressed genes. We hypothesized that maybe newer maize-specific genes or those that were dispensable across the maize genome might be more likely to be associated with differential expression, but once again, these distributions did not differ between DE and non-DE genes. And also, I don't have a figure for it, but it's worth mentioning that we did run a Go analysis to see if there were any functional characteristics enriched amongst either DE or non-DE genes. Once again, no notable Go categories popped out. And finally, slightly surprisingly, going back to the promoter SVs themselves, we just looked at whether and how the SV overlapped TE sequence. So we hypothesized that maybe having more of the SV made up of TE sequence might be associated with differential expression, or perhaps fully containing one or multiple TEs. One or more TEs within your SV might be more likely to be associated with differential expression. But we didn't really find a difference between DE and non-DE genes. And that doesn't mean we didn't find anything with TEs. Hold on to that thought for now. But generally speaking, we could not find any distinguishing characteristic of SVs that were associated with differential expression relative to those that were not associated, which suggests that there maybe aren't predictable features of SVs that result in differential expression. So finally, we wanted to focus exclusively on TE-mediated SVs. So those promoter SVs that had any overlap with TE sequence and see if there were any associations with differential expression. And while the amount of an SV made up of a TE sequence was not associated with differential expression, if we looked specifically at the superfamily of the overlapping TE, we did find an enrichment for LTR retrotransposons amongst genes that had differential expression between structural haplotypes. So LTR retrotransposons in the promoter were more likely to result in differential expression of the nearby gene. Furthermore, if we specifically looked at differentially expressed genes, we could ascertain whether presence of the SV sequence was associated with increased or decreased expression. And so for class 2 DNA transposons, we observed variable effects between the superfamilies. Some superfamilies were associated with reductions, while others were associated with increases in expression. Non-LTR retrotransposons, for which we only have one superfamily, were associated with increases in expressions. But interestingly, LTR retrotransposons were consistently associated with decreases in expression. So the only defining characteristic we could find of promoter SVs associated with differential expression had to do with the actual sequence content or superfamily of TE-mediated SVs, with LTR retrotransposons in the promoter being associated with differential expression, and in particular being associated with reductions in differential expression. And so kind of wrapping up what I've talked about so far, we do find that promoter SVs are associated with genome-wide changes in differential expression, and these changes appear to be context or tissue-specific in nature. However, there are very few defining features, basically no defining features of promoter SVs associated with differential expression. The only feature we could find had to do with TE-mediated SVs, where LTR retrotransposons were associated with reductions in expression. And so I've told you a lot about structural variation, particularly TE-mediated variation and its potential impact on global transcriptome variation. But how does this relate to what we are here for today, which is organismal adaptation? Well, as sessile organisms, plants need to have mechanisms to generate new variation in the genome and transcriptome in order to adapt to their environment, right? Plants can't just move to a better location if their environment becomes stressful. Their only tool is managing gene regulation in order to create multiple physiological responses from a single genotype. So in other words, plants are really masters of this adaptive phenotypic plasticity. And TE-mediated promoter SVs actually provide a mechanism by which plant genomes can do this. TE-mediated SVs contain regulatory features that are usually hidden by epigenetic silencing. But we know that the epigenome shifts in response to environmental stimuli, and we know that it's different across various contexts. So under environmental stress, the internal regulatory motifs within TEs could be hidden or exposed, allowing for adaptive changes in gene expression. And so something I think about a lot is how resilient host genomes are at responding to internal conflict, and how often they seem to turn lemons into lemonade. And this feels like a mechanism by which TEs could actually provide an advantage to host genomes in a way in which they could be co-opted or domesticated. And so while this study didn't explicitly look at stress response, we're really excited to look at some stress responsive assay data and see whether there's actually these TE-mediated SVs are driving a stress responsive or a stress response, differential expression as a stress response. There we go, I got there eventually. But with that, I'm going to wrap up. I just want to thank my collaborators, particularly Andy Reid, who's been an intellectual partner on this since day one, as well as all three of my advisors that I've worked with throughout the duration of this project, Nathan Springer, Yaniv Bronvane, and Candy Hirsch, as well as NSF for funding this project. And with that, I will happily take any questions.